Greetings, everyone, and welcome to today's seminar. I think this seminar is an extremely important one that we're going to be thinking about today. And salmon have been a key member of the coastal ecosystems that have sustained plant and animal communities for thousands of years along the BC coast. And undoubtedly, these are one of the reasons that the Coast Salish peoples were able to thrive along the coast for thousands and thousands of years. And it's for those reasons, again, that they established themselves here and why we're speaking from the traditional unceded territories of the Coast Salish people, in particular the Musqueam, the Stolo, the Tsleil-Waututh communities. And actually, I'm in the Neuse right now. And so on the traditional territories of the Snuneymuxw First Nations peoples. And these salmon, which have sustained these communities for thousands and thousands of years, are also been instrumental in the establishment of our own communities here in terms of when we came to these coastal environments and shared these communities with our First Nations peoples. And we also became entirely dependent on it. Many, many communities all along the coast. Myself, I commercially fished off a trawler when I was in high school. And I've experienced, as many of us experienced, the implications of the collapsing salmon populations along the coast and the kinds of effects that they've had on the entire ecosystems in terms of human communities, but also animal and plant communities as well. And so Gideon has been tackling these kinds of questions head on. He came to British Columbia initially from the UK. I had the privilege of meeting Gideon when I was at a meeting. I think it was in Plymouth in UK. And we talked about the possibility of Gideon coming to British Columbia and working on a project that we had just had funded to try and do viral discovery in Pacific salmon. And so Gideon had just finished up his PhD work in which he had looked at the diversity of viruses associated with the collapse of honeybees, honeybee colonies. And I thought, wow, this would be an absolutely great skill set that we could take work with him and his knowledge to try and understand if we could use similar kinds of approaches to uncovering viral pathogens that might be infecting salmon. And I was pretty skeptical that we would actually be successful at this. It seemed like a pretty big long shot at the time. But Gideon came in and using lots and lots of data that was provided by Christy Miller's group at DFO, uncovered many, many new viruses that we had absolutely no idea that were associated with salmon. And that looked like they might be really important, not only just that viruses were there, but pathogens as well. So Gideon made tremendous progress and went from there to be a Liber Aero postdoctoral fellow at the University of British Columbia, working again with Christy Miller and Dr. Jeff Joy in medicine, who's an epidemiologist, and now has just recently started at the Institute of Oceans and Fisheries, working with William Chung. So he'll be neighbors for us all. And it's my great pleasure today to introduce Gideon, and he'll be telling us about the work that he's been doing on viruses and wild Pacific salmon, and so which he's called the underwater epidemic. So Gideon, thanks very much, and we really look forward to hearing your talk.
Thanks, Curtis. Just going to share my screen here. Okay. How does that look? Okay. So yeah, I've entitled my talk "The Underwater Epidemic," and I'm going to explain why I use that term. And probably from Curtis's introduction, you already heard about emerging viruses, and I'm going to explain that as well. And my presentation kind of has three parts. I'm going to talk about some of the underexplored diversity that Curtis mentioned. Oh, sorry. And then I'm going to talk about a paper I published earlier this year, which is all about a virus called Piscine orthoreovirus, PRV, and how it's transmitted between farmed and wild populations. And then I'm going to get to some, the last part of my talk, I kind of get to policy and regulation, and going to kind of tackle DFO science review process and some of my experience with that, and kind of hopefully open up a bit of a discussion, because these are issues which don't only relate to aquaculture and salmon, but across all fisheries. And I think that would be of interest to everyone at the IOF. So yes, as Curtis mentioned, my project and all of my work since I've been in BC has been part of Christy Miller's strategic salmon health initiative. And this is a kind of big team of scientists, and the main aim was to try and work out if disease is an important factor in the early marine mortality of salmon, because we've identified the early marine stage as important potentially to salmon declines. So yeah, just to recognize that I'm part of this big team, and there's lots of different people I'm going to acknowledge at the end, and I'm going to share work for myself, but also a lot of others today as well. And as Curtis mentioned, salmon are a foundation species. They're really important not only to the ecology of BC, but also the economy and indigenous peoples. And they're in decline, and or certain populations in decline, and this decline has been pretty devastating for lots of different reasons. Just as an example, southern BC Chinook were assessed by COSWIC in 2018, and of the 16 populations, 8 were endangered, 4 were threatened, and it really was a not very happy picture. So the title of my talk is The Underwater Epidemic. And when I use the term epidemic, I use it, very consciously. An epidemic is when an infectious agent grows beyond its normal level. So this is what we're kind of used to thinking about as an outbreak or an outbreak dynamic. So this is when a virus or an infectious agent grows. And it's the opposite of an endemic disease, which is a disease which is kind of stable within a population. And as we've been experiencing the global epidemic or the pandemic of SARS-CoV-2, a lot of us are becoming more familiar with these words, which is kind of useful for me. And here I use another term, emerging viruses. And emerging viruses are a bit more straightforward to explain. They're basically just a virus which is newly discovered or new to a region, and it's increasing in incidence, or just has the potential to increase in incidence. So as Curtis mentioned, I've been lucky enough to study viruses in a few different systems now. I started out working on viruses that infect phytoplankton, and some of you might recognize the coccolithophore on the left there. For my PhD, I worked on viruses in honeybees and other pollinators, and was really interesting working with this vector of disease, which is a small parasitic mite called Varroa. And then for the last 5 years, I've been working on viruses in salmon. And the thing that really links all these together for me is the molecular tools that we can use to ask questions about these viruses. And we're kind of a little bit journalistic. We use the 5 Ws. It's who's infecting who, what, where, why, and where, how is that transmission happening, and even when. And I'm going to get to the when a little bit in my talk as well. And viruses play all sorts of different roles. They're really important in terms of driving global cycles, as you might have heard from some of Curtis's work at UBC. They're everywhere, and they modulate and the function and evolution of all life. And we're surrounded by them all the time. And it's only in certain cases where they become more of a problem. And I think viruses are really interesting to consider in salmon, because we know that when population sizes get small, viruses can become more important. And it's this kind of idea of using virology within conservation, I think is really exciting field, and it's growing very quickly. This is a kind of probably not used to having such furry examples in the IROF, and there's probably, it's going to be more fishy later on in my talk, but this is an interesting case study of the Ethiopian wolf. It's the rarest canid species in the world and the most threatened carnivore in Africa. And you can see from this plot that its populations dynamics are largely controlled by viral outbreaks of different viruses in that population. And wildlife managers are going in and vaccinating and trying to mitigate some of these changes. But when we come to aquatic viruses and viruses in fish, we actually don't really know much about them. This was a study in 2018, which just in this one study discovered hundreds of new viruses in fish and aquatic vertebrates. The plot shows the number of RNA viral species, and you can see that in mammals, we know of many, many species, and in fish, we know very, very little. And this, I think, is why the discovery part of my work was quite interesting. I'm going to touch upon just one example. We discovered, over 15 viruses and counting new species, which many of which we'd shown to infect salmon. But I think this example was really interesting because it was the first corona-like virus to ever be discovered in fish. And it's evolutionary related to mammalian coronaviruses, which obviously now we all know what they are. And what was really interesting with this virus is that when Emiliano De Chico, who's a veterinary pathologist within the Christie's group, when he looked where this virus was infecting, we localized it to the gills. So we knew that this virus, a lot of these viruses in mammals infect the respiratory tissue, and in salmon, we saw the same thing. And the plot on the right shows the prevalence of this virus in different populations. And what was really interesting was this virus seemed to mainly infect Chinook, and we discovered it actually associated with a lot of salmon enhancement hatcheries. Many of you will know that these salmon enhancement hatcheries augment wild fisheries. So we found it in several hatcheries, not all of them. But what was really interesting was that we found that this virus, we detected it whilst in hatchery fish in fresh water. And it was in the gills as these fish undergo smoltification. And these are changes which the gill tissue has to undergo as those salmon prepare to enter the marine environment. So we found it was highly prevalent during the freshwater stage. And once these fish were released from hatcheries, we could detect it in the marine environment, but not for very long. And soon after, that signal just disappears. So this really raises the question as what is this virus doing? And is it associated with, is it the cause, or is it just associated with this decline in another way, or is it just not related at all? Like we don't know. And this is something which, my project's going to continue working on for the next few years, and something I'll be working on while I'm here at the IOF. So just to introduce this idea, that the concept that not all viruses cause disease, and for disease to happen, which is represented in red in this plot, it's a combination of the agent of a virus, the environment, and the host. So it's actually a very complicated system that we're working on, and different people in Christie's labs are working on different kind of aspects of this, what is known as the disease triangle. And not all viruses cause disease, some do, and I thought I better touch upon what this means in terms of fisheries. These photos are from a manuscript which described a viral outbreak of a virus called VHSV. This is a highly contagious virus which leads to high mortality in a variety of different species, and in this example in Pacific herring. And you can see that you get these overt kind of outbreaks of disease. You can see fish which obviously are not looking very healthy. You can see die-offs. But this is not always the case. And studying disease or viruses and disease in wild populations is tricky. And my feeling is that most instances of disease in wild fish is mostly covert. And by that, I mean, it's things that we don't see, and there's multiple reasons why we don't see them. But one of them is that we know that fish which have disease are picked off by predators. And there's a great study from UBC, which came out this year, which showed that in the stomach of bull trout, juvenile sockeye, which were predated, were more likely to be infected with a virus than those which were not predated. So we know that predation is part of this. And that's why we might not expect to see these kind of late stages of disease like we see in the herring in all wild fish all the time. So studying, as I said, studying disease in wild fish is complicated, and it's pretty rare that we see wild fish die, and very rare that you would expect to see very severely diseased fish just swimming around in the wild. And for that reason, we think these kind of sublethal effects. So we're not only using mortality as kind of a bottom line, we have to think about things that might affect the fish in other ways. And I kind of knew that this was going to be important for my work on honeybees, because the honeybee virus I worked on wasn't the most virulent of the viruses out there, but it was the one which was the most common. And actually, in some cases, survival was affected just by changing the behavior of the honeybee. So just changing the behavior could be small enough to lead to poor survival in a wild animal. And much of my work revolves around viruses and aquaculture. And I think there's lots of different reasons why this is really interesting, but these open net pens can actually be really great places to study disease. Some of the work we've done on Chinook is informed by farmed Chinook, because in the wild, we don't see these late stage of disease. We see lower prevalences of the viruses we're studying. And we can get samples of later stages of disease. And I'm going to get to this a little bit later in my talk. And I'm sure all of you are familiar with salmon farming in BC. It's a huge industry in Canada. And a really important kind of thing to think about is that most of the farmed salmon, rather than Chinook, like I just mentioned in my last slide, most of the salmon farms in BC are Atlantic salmon. So this is a different species from the Atlantic, which was introduced over a long period of time to BC, and more recently, in the last 30 years, has been farmed quite intensively. So just touching on this idea that, and this is not unusual to marine aquaculture, and the same thing happens on land, is that as humans, we've intensified all of our farming activities. And actually, as well as studying what this means for human kind of evolution and where we're going as humans on this planet, it's actually a story of disease emergence. And a lot of the study of disease is in farmed animals or farmed crops, because as farming gets intensified and you put lots of the same species in one space, it's actually the perfect place for diseases to emerge. And aquaculture is no different. We move species around the world. We bring non-native species into new regions, and this can bring new species into hosts, which they're into kind of neighbors they've never met before, and that goes both ways. And then we put them in these very high density net pens. And obviously this high density, we understand this from COVID-19, is a good place for favoring outbreak dynamics. And then you add to that in the marine environment, viruses can freely float in and out of these net pens and can be dispersed within the water very easily, and they've evolved for a long time to be able to do that. And the idea that as you have a high density, you have more pressure to evolve virulence. It's kind of no surprise when you put all these factors together that we do see emerging viruses in aquaculture, and that's something that we've seen all around the world in all different systems. So I'm going to talk about the virus called Piscine orthoreovirus, and it's a virus some of you might have read about in the media. It is a paper which I published in earlier this year. And I'm just going to give you a very brief history of PRV. In the late 90s, there's these outbreaks of a heart disease in farmed Atlantic salmon in Norway. And a virus was suspected, and it wasn't until 10 years later that that virus was actually identified. And when we look back, it's actually quite a long time. But that's kind of how these things go. Sometimes it takes a long time to link new viruses to the disease, which we now know they cause. In 2013 was the first genomic sequencing we saw of this virus from BC. And it looked similar to viruses in the virus in Norway, but there's been a lot of contention about where this virus really came from, how long it's been here, whether it causes disease or not. And I'm going to try and get to some of those issues with with all the data I'm going to present to you today. And one of the kind of key questions that a lot of people have and key concerns, and really a growing concern, is that the diseases are being transmitted from these open water net pens to the wild juvenile salmon which swim right past them. And probably this video, which is very eye-catching, is kind of a good example of ways disease transmission might happen. This is a video taken of effluent from a salmon processing plant, and the blood was collected, and PRV, the virus, was in that blood. And it's just the idea that transmission is possible. I was at a fish health meeting in 2019, and scientists and government representatives were saying, yeah, but there's no evidence for transmission of PRV between farmed and wild salmon, which was maybe there wasn't a huge amount of evidence. And I thought, well, using the kind of genome sequencing that I like to use in my work, we can actually get to that question. So we designed a study to investigate this. And we can do this because viruses leave a genetic fingerprint. We're all used to kind of the idea of having a PCR test for a virus, and that's something which Christie's lab has done many, many times. I'm going to show you some of that data. But we can also sequence the genome of the viruses. And using those genome sequences, we can test the transmission between different populations. And we can get to this question of, well, where does the virus originate? And we can also use those sequences to get at some other questions like, well, how has this virus population changed over time? So Christie's group has been, and like a lot of these samples come from other groups within DFO, sampling migratory juvenile salmon throughout southern BC for over a decade. And this just shows the number of samples in the different regions. So you can see the sample size is quite high. And Christie's lab has been testing many of these samples for many different infectious agents, not just PRV. But I knew that this was a really kind of rich data set to use to get at some of these questions. So in the study, we actually have access to over 10,000 fish which have been tested for this virus. And the circles on this plot show the samples which we selected for sequencing. So it's really cool to get to work on a project where we have such a rich, large data set. And as well as looking at wild fish in this paper, we had data from farm fish, and these were data from the aquaculture audit. And what these data show is that PRV is very, very common on Atlantic salmon farms. Over the period of a grow-out cycle on an Atlantic farm, the probability of those Atlantic salmon becoming infected with PRV becomes higher and higher, until by the end of the cycle, you're pretty much, it's very, very likely that that fish is going to be infected with PRV. And kind of my specialism for this project was using the viral genome sequencing to get at this transmission history. Because RNA viruses have quite high mutation rates, we can look at how changes in these viruses over time tell the story of the emergence and transmission. And I'm going to show you just 2 phylogenetic trees today, so it shouldn't be too heavy. And the analysis I used, it uses timescale trees. So actually, in this example here, we're moving from the past to the present. And you might be familiar with some of these kind of tree diagrams. They've been used a lot to track transmission of variants of SARS-CoV-2 around the world. And we can do this because as those viruses are transmitted, there are mutations which we can track to look at changes in the genomes. And combining these with the locations and timings of where things were sampled, and throwing a few mathematical models at that, we can actually infer the timing of these changes that happened between different locations. So I know there's a lot to look at this slide, and the details of it aren't necessarily really important, but I want to show you how I came to the conclusions that I came to. So here's a timescale tree of many hundreds of PRV samples, again scaled by time. So the most recent samples are at the top on this tree, and we can see over time there's certain things that happened, and we have lineages of PRV which have diverged. The color of these branches represents different regions around the world. And I know this is a lot to look at, so I just took all the kind of important bits of information from that tree and put it onto a map. And what we actually see is that the origin of a virus, the origin of PRV from all the sequences, is in Norway. And there are several events on the tree where the virus was transmitted around the world. And we can see that there's been a transmission, for example, about 30 years ago to British Columbia. But there's actually other introduction events. So we know there's a more recent introduction event of a virus via Iceland into Washington just in the last decade or so. But interestingly, there's also introductions to other parts of the world where we have sequencing from. And in Chile, for example, where they farm both Atlantic salmon introduced from Norway and Chinook salmon introduced from the Pacific, they have two different lineages of PRV than they've been introduced from either side of the world. So I think it's really important method to get at what the situation is now. We can use the data from now to infer what's happened in the past. And we probably only see, because we only have a certain amount of data, we're probably only seeing a selection of these introductions. There's probably been several. Into BC, we know of at least 2 introductions. I'm sure there's been more over the years. But the virus which we see circulating in BC, we estimate was introduced about 30 years ago from Norway. So that's kind of some of the genomic sequencing we use to look at the transmission around the world. But using all those genome detections, all those PCR detections of PRV, we can look at the epidemiology. And we're really interested in obviously getting at this idea of this question of transmission. So we kind of looked at the data in a way where we could ask, well, is transmission happening from the net pens to the wild fish? How can we look at this? We know the virus is on the farms. We've shown that. What about the virus in the wild salmon? Do we see more of this virus in wild salmon which are closer to farms? And for this, we broke down our data into 2 halves because we have different types of, and this analysis is all on Chinook salmon. And we have different populations of Chinook salmon in our study region. In the spring and summer, most of the Chinook salmon we sampled were from the Columbia River. And in these, we see no relationship in the proportion of PRV and the distance they were sampled from active farms. In the Folland Winter, most of the fish in our database were from rivers in BC. And in the Folland Winter, in these Chinook salmon, we do see this relationship. The closer the fish were sampled to active aquaculture, the more likely they were to be infected with this virus, PRV. What was kind of interesting is, and I'm going to touch upon this in the next slide, is that in these 2 different populations of Chinook salmon, we actually see slightly different lineages of PRV. So in the Chinook salmon from the rivers in BC, we see the same viral variants in the wild salmon as we do in the farms. But in the salmon from the Columbia River, we see a slightly different lineage. So it was kind of neat that our genomic data and our epidemiological data came to the same conclusion. And this is another analysis where we are looking at these different populations of salmon and the lineages of viruses within them. And basically what we see is that the viruses that were sampled from these BC wild salmon in orange, these were kind of interdigitated in the tree within the dark blue here, which is the farmed Atlantic. So this is further evidence of this transmission. Whereas the yellow and blue here are the Columbia River fish. And actually, so those Columbia River fish, which have that slightly different lineage, it's similar to a lineage we see in the Chinook farms. So there's potentially something else going on there. So yeah, we have these 2 different lines of evidence for this transmission that's happening. We can also use those genome sequences to look at past population dynamics. So because those changes in viral genome sequence happen at a known rate, we can look at those changes over time to try and get at the past population dynamics of that virus. And what we see is the shape of the curve here represents the number of infected fish which go on to infect additional fish over time. And we see this expansion in this infected population size. And I think this wasn't necessarily a surprise, because we see the same kind of expansion of infection on farms on every Atlantic salmon farming cycle. And interestingly, there's another study using a very similar technique on another salmon virus in the Pacific here called IHNV. And this is a virus which is endemic to a region in sockeye. And their analysis, the line was pretty much flat. They don't see this kind of change. So this is where I'm getting to the idea of like, well, okay, do we have an endemic virus which is stable in population size? Or are we seeing something akin to an outbreak where that growth in population, the size of the population is growing over time? We see that when we use this analysis, and we see that same growth when we look on individual farms. Of course, a really important question is, well, does this virus cause disease? So PRV is known to cause heart disease, a disease called HSMI, or heart and skeletal muscle inflammation, in Atlantic salmon. But the important question to Pacific salmon is, well, what disease does it cause a disease in Pacific salmon? The ability for it to cause disease in Atlantic salmon doesn't necessarily predict what the impact is going to be in different species. Some of Emiliano's work has shown that PRV is associated with a different disease in Chinook salmon, and he found this by studying farm Chinook salmon. And that's why I mentioned these farms are kind of useful indicators of what might be happening in the wild. He showed that PRV is associated with this virus, with this disease called jaundice anemia. And this is a disease where, as a result of the infection of the red blood cells, the red blood cells lyse or burst open, and this causes disease damage to the kidney and the liver. And in severe cases, this can lead to this yellow in coloring. And it's not just viruses, it's not just PRVs that's associated with this farms. I want to touch upon this work very quickly, and I think it will be clear why. This is Tenacibaculum is a bacteria which causes disease in Atlantic and Pacific salmon. And this is a study which a colleague of mine recently published, which shows that as wild juvenile sockeye swim out the Fraser River and migrate north, the prevalence of this bacteria peaks where the farms are. So I just want to quickly introduce that bacteria, because I want to get to the next part of my talk, where another colleague of mine has carried out a study to ask whether some of these pathogens which we're looking at in wild salmon, whether they actually have an effect on populations. And how he did this study was using this data set where we've looked at all these different infectious agents in wild salmon. He used these data to model if pathogen prevalence has an impact on either survival, and I'm not going to go into details, but there's ways which we can know the survival of various populations, and body condition. So deviations from this mass length relationship which fish have. So basically, are certain pathogens associated with skinniness of a fish or poor survival? And when he did this analysis, I almost couldn't believe the results, because the two agents which came out on top, so this plot just shows the posterior probabilities of an association being negative for either the survival or the skinniness in Chinook or coho in different seasons. And the two agents which came out on top were Tenacibaculum and PRV. So it's kind of interesting that these two agents, which we know are associated with the net pens, came out on top. And at first I was like, well, this is really bad news, because these are agents which are associated with farms, and they're potentially causing, an issue on the populations of wild salmon. But on the flip side, this suggests that there are interventions which we might be able to do to prevent this, because these are agents which via these net pens, we are potentially adding to the amount of infection in wild fish. So I think this is a really important correlation, which is been shown, is currently in review, and that's work from a colleague called Art Bass. So that's kind of the sciencey part of my talk, and I can see I've got a few minutes left to I wanted to touch on something people always ask me about my work is like, okay, well, you found this, what's going to happen now? And that's kind of not the role of scientists, it's the kind of role of decision makers. So I'm going to skip this conclusion slide, and I'm going to go straight to the next question, the final part of my talk. When I speak to people, they ask if my research has influenced how PRV is regulated on farms. And the answer is not really, because DFO currently do not consider PRV a disease agent. And this is kind of a snippet from the form which DFO have to decide whether a virus is a disease agent or not. And you can see, like, the first question is, has the infectious agent been shown to be associated with disease? Yes, and there's a bunch of discussion of that. And the second question is, has the infectious agent been demonstrated to cause disease in Canada? And here the language is interesting, because it's a cause means we have to have a causal relationship, and in Canada means it has to have been done in Canada. The isolate of PRV, or the lineage of PRV, which is in Canada, has been shown to cause disease in Atlantic salmon, but the study's been done in Norway. So in this study, they tested all these different isolates of PRV, and they found that they all differed in severity of disease. The Canadian isolate only caused mild to moderate disease, but it still caused disease compared to the controls, and the important thing. So I think the way which policymakers decide whether a virus should be regulated or not is a question which is, for the scientists, it's actually, we need to be involved in this discussion, because to me, there's no doubt that this virus does cause disease. Whether it causes disease in Canada or Norway, it's kind of irrelevant to the question. And actually, what we're worried about here is whether it causes disease in Pacific salmon, not in Atlantic salmon, and that's a whole nother question in itself. I'm going to show some slides of a kind of another snippet on the kind of policy side of things. And this summer, there's a standing committee on fisheries and oceans, where this MP, Gord Johns, who's an MP on the island, I hope maybe he read my paper, which was published in May, and he asked the Ministry of Fisheries, how will you evaluate the implications that this virus, which has been imported and is spreading via the farms, how are you going to evaluate it? Will you be designated PRV a disease agent, because there is mounting evidence that it does cause disease, and it has to be designated a disease agent to be captured under the policy. And the minister's answer was quite striking to me. I was watching this live and I was quite surprised, because her answer was basically, we're very interested in new research, but all of our science is peer reviewed, and we'll do our peer review process, and we have the best peer review. And for me, this was quite striking, because as scientists, of course, we're used to our work being peer reviewed, but we don't use peer review as a way to block new research that's coming out. It's not like oh, well, we'll have to do our own peer review of that work, so we're not going to worry about it. And that was kind of the answer that we got. And Mr. Gord Johns pointed out that maybe we need someone outside of the bureaucracy and political system to conduct these reviews. And I'm going to get at what he's talking about there. And it's not just me and Mr. Johns who are worried about the peer review process within DFO. And I kind of want to speak about this with the IOF people today, because this is not just a virus or not just an aquaculture question. This is relevant to all fisheries. And it's not just me who's saying this. The Chief Science Advisor of Canada, in her report on aquaculture science, she's recommending an external peer review and an external advisory committee, external to the bureaucrats within DFO. So I've been kind of diving into the way DFO review science, and I can see that I'm running out of time, and I want to leave time for questions, but I'm going to very briefly kind of introduce this is how DFO this is their formal peer review process. Some of you might have heard it. It's called the CSAS process. And I was kind of looking into how it's done, and it's so different to what we're used to as scientists. I think it's worth touching on some of the flaws. Basically, one of the ways DFO carries out these assessments is that they have to choose what the questions are. And there's a real problem with selection bias, in my opinion, in this process. And a real good example is that for the risk assessments on the risk that pathogens associated with farms in the Discovery Islands pose, there were meant to be 10 risk assessments. Then there were 9, and the assessment for sea lice was just missing. And then they decided that these pathogens cause a minimal risk. We didn't even look at one of the pathogens. And there's similar kind of issues within the CSAS process where reviewers are demanding the removal of certain questions because they don't think they're pertinent. And in 2015, and I've got all the evidence, if anyone doesn't believe me, I can show you the paperwork. In 2015, a reviewer requested that we remove the issue of whether PRV caused a disease from the objectives of that review because they said it wasn't important. Confirmation bias is the idea that we have a tendency to interpret new evidence as that align with our own beliefs. And this is a concern because the reviewing panel of the science is not truly independent, and there has been until recently no way for anyone to state their conflict of interests. And my point here is the reviewing panel was made up of industry representatives who maybe wouldn't be entirely impartial when it came to assessing the kind of evidence I've shown you today. And this is important because the chair of the review chooses who is on the reviewing panel, and then it's a consensus-based review. So I'm going to have to move through this quickly so we have time for questions. There's within this process, you can't make anonymous comments. So there's little incentive to be kind of critical as you might be if you're reviewing a paper anonymously. And I'm just going to skip this last point. So I know that these examples are not just restricted to aquaculture. This was a story in the Narwhal earlier this year, which found a DFO report within the Species at Risk Act. So not CSAS, but another way which we use science to make decisions. And here the report was edited by managers to downplay the risk to steelhead. So I'm really interested to hear from other people within the department if there's any other examples within other fisheries you'd be willing to share where you think, oh, this review process is letting us down. Because at the end of the day, we all do this really important science. And if there's not a fair way for it to be incorporated into the decision-making process, I think we all know that's a problem. And I don't have a problem with being an advocate for good science and good impartial reviewing of science. So I think I'll end my talk there. I just want to acknowledge all the different people who, and I'm sure I've missed some off, who've been involved in this work. The funding for my, or 50% of the funding for my new position is from the Pacific Salmon Foundation, and my past project was from the Libero Fellowship. And a special thank you to the photographers who lent me their images and videos to use in my presentation. And yeah, I'd like to open up for discussion and questions.
Thank you, Gideon. I think we have a first question from Andrea. Andrea, if you want to ask your question.
Thanks very much, Gabriel. Thank you, Gideon, for a great talk. And yeah, I want to commend you for, really acknowledging the social context in which science and scientific processes sit. So thank you for that. I have one technical question and then one maybe more institutional or procedural one. The technical one is the one that's in the chat. And I'm just curious about if there's also a freshwater source of PRV or if it's seen as a strictly marine sourced disease.
So yeah, we do detect PRV in freshwater, and we've detected PRV in the freshwater hatcheries which stock Atlantic salmon farms. And I have the feeling that there might be PRV within the Columbia River hatcheries, and that's why we see this distinct lineage there. Although I don't have any direct PCR, I haven't seen any data from those hatcheries themselves, but the sequencing kind of suggests that might be happening. Does that should answer your question?
Yeah.
Sorry, in the second half of your question.
The second half, I'm just curious, you showed the map from where all of the samples that you're working with are coming from. And, we're looking at indigenous territories across this province. And I'm just curious whether there's efforts to share the outcomes of this work back with nations. And if not, we should have a chat. And I'd love to talk how we could do that.
I think you can never do enough of that. So yeah, I'd like to have a chat with that. Last week, I presented at the BC Council of Indian Chiefs. And this week, I presented some of this work to the First Nations Summit. But I'd like to do more of that kind of communications. I think it's especially important as there's a First Nation operated lab, which is, could be doing this kind of work. And the kind of sharing of these technologies and this kind of work so that nations can use this kind of work within their own territories, I think is really important. So yeah, I should also plug a colleague of mine is giving a workshop on eDNA, and there's funding for Indigenous youth to come and learn how to use that eDNA. So yeah, I'm plugging. If someone would like to know more about that, please get in touch as well.
Thank you, Gideon.
I think we have a question from Tony Farrell. Tony, if you want to ask your question, please unmute. Okay, so I will read it. So Tony Farrell was asking you, your model apparently does not account for the billion of Atlantic salmon were introduced into BC waters over 100 years ago. How do you know that PRV was not introduced then?
It's a good question. We don't know, we can't say for sure that it wasn't introduced then, but what we can say is that the PRV that we're detecting in the farms and the wild fish now is so similar to what it is in Norway that we don't think it was, and the sequence, it would be kind of impossible for the sequence similarity to be so close with the modern day Norway lineage. But of course there could have been multiple, and I kind of got to that, the idea that there could be multiple introductions and we don't have the data to have, we don't have sequences from those samples. The other thing I'll say is that because we know there's multiple introductions, we know there's an introduction about 30 years ago. We've got evidence for another more recent introduction in the last decade. So even if there were earlier introductions, the lineages which we see now are probably more likely to be a result of those later introductions.
Thank you, Gideon. I think we have a question from Rick Taylor. Rick.
Yeah, thanks. Yeah, Gideon, excellent talk. Thank you very much for that. Just a couple of comments rather than a question. First of all, to me, the issue of when it was first introduced is kind of irrelevant, because it's clear that the problem, whether they're introduced 100 years ago or slightly more recently, that is being amplified by the farms. The second point I wanted to make is that, again, you and I have talked about some of the work that purports to show no effect of PRV on some species of Pacific salmon. But even if the effect, which we can discuss, but even if the effect is minor, it's important to keep in mind that the whole idea of cumulative effects. So the last thing Pacific salmon need, even if it's only 5% mortality, is yet another threat towards them, given all the other threats that they're facing. The third point I want to make to emphasize some of the things your talk is this point about the independence, the lack of independence, and the conflicted mandate of DFO is extremely important. And not only are you raising it, but the Royal Society of Canada, in their last report on sort of review of Canadian fisheries, came out with a very explicit statement that DFO clearly has a conflicted mandate and needs some much better external oversight. And I know that the Auditor General's Office of the federal government is compiling information right now to see if a full sort of investigation of how DFO does its work, and particularly in terms of the lack of external review in many cases, is ongoing. So hopefully the Auditor General, once the sort of bureaucracy of the Auditor General's office gets up to speed or gets all the information, the Auditor General will make a recommendation to do a full investigation, particularly focusing on the lack of independence. And the last point to emphasize this is this was also a recommendation of the Cohen Commission, which has not yet been implemented, sort of oversight over how DFO in this particular case has independent oversight of how DFO has or has not implemented the 75 recommendations of the Cohen Commission. And this is extremely important because DFO's own assessment says they have implemented 100% of them, which is clearly not true. So again, thanks for your talk.
Thanks for those comments. I guess I just kind of echo your last point on the reviewing process there and just say, this is something as scientists we should all be talking about and discussing and maybe even grouping together and, you know, pointing out where these flaws are. I think if we just sit back and expect the kind of powers to be to do their audit and fix everything without being outspoken scientists ourselves, I'm concerned that it will just be another kind of box ticking exercise. And yeah, this is something I'm kind of actively working on. So if people want to get in touch with those kind of questions or points, I know the aquaculture kind of file very well, but I know there are examples from outside of aquaculture which I'm interested in hearing about.
Thank you, Gideon. I think we have a question from Curtis.
Yeah, hi, Gideon. Thanks very much. I love hearing what you've been up to and also the context that you put your work in, I think is fantastic and very appropriate. And the question that I had is, of course, one that you're really familiar with, and I don't know, but what the answer is, and would like to hear your perspective. And you know, as you pointed out, viruses cause sublethal effects. In fact, almost all viruses cause sublethal effects, right? So it's very few things that cause outright death. I think, the 140 viruses that infect humans, almost none of them cause death most of the time, right? So it doesn't mean they don't make you sick. So how do we get from the question, not only with PRV, but all the viruses that you've found, how do you get to this, like, what's the effect of each virus or any virus on ocean survival, right? That's the one where we have the most contention, because it's people, there's certainly industry will argue, well, you haven't demonstrated that there's any impact on survival. And I think how do we get there?
I think, we'll only ever get so far, because we'll never be able to show that, or it's really hard to show that in a kind of cause and effect relationship. All we have right now are a kind of associations, correlations between detection of infectious agents and this kind of skinniness or poor survival. The only way you'd ever be able to show a cause and effect would be some sort of challenge and release, whether that would be, you vaccinate some fish against the virus and release them and see if they survive better. There was a work which I know of someone who was trying to do that with sea lice, and they didn't get permission to do that work from DFO. But you're right, because we're working, and I'm sure a lot of the fishery scientists here are used to that, it's always going to be correlative, because we don't have a control planet to do these experiments on, really. So I think we have to use all these different molecular tools, whether it's the sequencing or the localization, the pathology, to have a look at what's happening within the tissues and the cells, the surveillance, and then all these models to look at the correlations. You kind of have to put them all together, as well as the lab trials. The lab trials are important as well, but you'll never, it's very, very hard to get to that kind of cause and effect relationship. But the good news is that within the legal kind of framework, we don't need to have a cause and effect relationship between, for example, a virus and disease or harm to a population to act. Written within Canadian law is the fact that the precautionary principle allows there to be some uncertainty and allows it to be a correlation and still make a decision. Where you kind of draw a line there, I think, is the really difficult thing to do.
Thanks.
Thank you, Gideon. I think we have a short question from Colette.
Indeed. I have lots, but I'll stick to the easy one since we're running out of time, or the shortest one. I was just wondering, I really like the way that you started just by making sure that we all understand context and also the definitions of certain terms that you use. And so I was just wondering, in terms of disease, since that seems to be like a critical word, or what is critical in terms of determining effect, is there a clear definition on what disease represents and what Canada or DFO understands as disease?
There are clear definitions which scientists use, and you have to define as you said, I like to define what I'm meaning when I say things. And there are clear definitions of disease, and these are just changes in the structure or function of a tissue. There's a whole nother example of contention around this issue of disease, and in Canada, they only diagnose disease when they see clinical signs of disease. So these are outward signs of disease you might be able to just observe on a farm, rather than these kind of disease changes to a tissue. But how DFO diagnose disease in Canada is not typical of how disease is diagnosed in this context around the world. So I think that's important to point out too. So yeah, it's a really good question, and we must define all these different words when we use them. But for me, the importance of that question is, if these even if the changes are mild, even if a disease is just mild, for example, heart inflammation, if that can have big consequences at the population level, then it's still something we need to consider. And it doesn't matter that we don't have mortality at a certain level. It's kind of irrelevant to the health of wild salmon.
Absolutely. Thanks, Gideon.
Yeah, thanks everyone very much for attending. I really appreciate it. I thought it was an excellent seminar and a perfect fit for many of the things that we think about in IOF, but also across the regulatory landscape, right? So I think this is one of the really wonderful things about IOF and the kinds of people that work there. They have people like you that are talking, but it's this linkages between kind of policy, science, and I think most importantly is that intersection with communities and peoples, right? So I think what we really recognize in the IOF, that these things are all so intimately connected. And I think that's one of the great strengths of not only IOF, but also the kinds of people that are brought in for seminars and also as new researchers. So welcome to the IOF as a new member, and I think you'll find it a great group of people, and the kinds of conversations that I've had are unlike any other conversations you'll have in a unit at UBC. I think it's a wonderful collection of individuals thinking about problems in very, very many different ways. Thanks, Gideon, and thanks everyone for attending, and I hope to see you at many more seminars as we go forward.